So let's begin. Sinew of sinew, when you sing a new tune of when you grew anew? Yes, muscle tissue. All right, let's reprise this guy Avicenna. I've talked about him a few times, and I'll just read you a translated quote from one of his texts. The power of locomotion is that which contracts and relaxes the muscles, whereby the members and joints are moved, extended, or flexed. Their power reaches the limbs by way of the nerves, and there are as many forms of power as there are of movement. Each muscle has its own peculiar purpose and obeys the decree of composite sense. All right. So what I think is, there's a couple things interesting in this quote to me. First of all, he, at this 11th century stage, understands that it is, in fact, muscles which are acting in this contractile fashion to move, to work on the skeleton and move the joints. And more importantly, that this is under the control of the nerves, that there's this relationship between the nerves and the muscles, the two excitatory types of tissue that are going to elicit this contraction. And then lastly, what I find really interesting is this has its own peculiar purpose and obeys the decree of composite sense. This is sort of the, like, nascent beginnings of the structure-function paradigm that it took Louis Sullivan 700 years to more fully articulate. So that's kind of an interesting thing there, this peculiar purpose, the unique purpose, and then the composite sense, the function and structure relationship being articulated by this early mind from Persia. All right. And then our understanding of muscle groups and their distribution was further quite a bit more by Salience. I've mentioned him previously as well. And this is really when our modern understanding of anatomy began, right? Because we weren't able to do careful anatomical dissections until this point. So all of this begins right here, what you're looking at, the neuromuscular junction. This is where nerves meet muscle. This is where one type of excitable membrane meets another at a point of synapse. We specifically call this the motor end plate, this neuromuscular junction. So in red here are muscle fibers, and in the chartreuse are the telodendria terminating from an axon terminating on the membrane, the sarcolemma of the muscle cells. We're going to talk about how this structure works in molecular detail throughout the lecture today. Okay, so to begin though, let's just talk about muscle tissue in general. It is one of the primary tissue types. I told you that a while ago. And there's three categories: cardiac muscle, skeletal, and smooth muscle. The skeletal muscle system is only made up of skeletal muscles. You can recognize it because skeletal muscle is the only muscle type that is voluntary. You do not have any muscles in your body that you can control with your mind voluntarily that are not skeletal muscles. All right, it's all skeletal muscle. And then morphologically, when you look at them, they are striated. They have these characteristic stripes, and which we'll come to understand in a little bit. We'll explore what that is. And they're multinucleate. They have many nuclei. Then are the two involuntary categories. So these are muscle fibers that you do not consciously cause to contract. And first, the cardiac muscle looks a lot like skeletal muscle, except that the cells are much smaller. So skeletal muscles can be quite huge. And they are mononucleate. So they are striated like skeletal muscle, but they're only one nuclei per cardiac muscle cell. And then smooth muscle cells really look quite different than the other two categories. They are mononucleate like the cardiac muscle, but they do not have striations. And I won't really talk about how they work on a molecular level, but there are some overlaps in how they work, but it's not in they have slightly different functions, and because of that, the molecular machinery for contraction is deployed a little bit differently. It's the same actors, but it's arranged a little bit differently in those cells. Okay. So there are some analogies between muscle and nerve. All right, we haven't really gotten into nerve tissue yet. We will, but there are some analogies between them, and this is one of them, talking about the connective tissue that supports muscles and muscle tissue. In a given muscle, there are going to be 3 layers of connective tissue. Likewise, in a nerve, there are going to be 3 layers of connective tissue. Here, in a muscle, the first is the epimysium. So the epimysium is this sleeve of connective tissue that surrounds an entire muscle. It's made of collagen. It's quite strong and has a connection to the deep fascia of the body. The main job of the epimysium is to give that muscle its own unique definition. It defines the muscle, it separates it from other muscles, and allows that muscle to slide in relationship to adjacent muscles. The next layer is called perimysium. Now, the actual muscle tissue itself is broken up into these bundles called fascicles. Fascicles. So, for example, here is the muscle taken from this person's forearm, and then if we look at it, this muscle has these individual fascicles. This would be one fascicle. A fascicle is itself surrounded by a layer of connective tissue called the perimysium. Now, perimysium is kind of the supply roads. It's like it's where all of the blood vessels and nerves that supply a muscle run. They run along the connective tissue that is the perimysium that surrounds each of these fascicles. And then a fascicle is itself made up of individual muscle fibers. A muscle fiber is a single cell. And each of these single cells are themselves surrounded by a layer of connective tissue called endomysium. And this is where the arterioles and venules and minor nerves are broken up into capillaries and individual nerve fibers, which will connect with nerves at the neuromuscular junction. This is where we find myosatellite cells. So the myosatellite cells are stem cells that live inside of a fascicle, and their job is simply repair damage that might happen to a torn muscle cell. Myosatellite cells. Yes, sir?
What's the point of?
We're going to get there. Great question. You just hold on to that one. That's the whole point of lecture today. So a couple things about skeletal muscle fibers. They're exceptionally long and large. Some of them can be as much as 30 centimeters long. That's like that big. A cell in your body that big. Holy smokes, it's alive. Woo. Yeah, very long and large. And this is because muscle cells develop through a fusion of cells that are of, I didn't talk about embryology, but there are 3 layers of tissue embryologically, and mesoderm is one of them. They're mesodermally derived cells, a specific lineage called myoblasts. Myoblast. At this point, you should be able to take that word apart. Myo, meaning muscle. Myo means muscle. And a blast is simply a not terminally differentiated cell, right? So like we had osteoblasts, there's like semi-differentiated stem cells that were then able to mature into the osteocyte. So myoblast is not a terminally different, is not a fully mature muscle cell, but they fuse, and in that fusion, they bring their nuclei with them. So when you look at an actual muscle fiber, you see that it can have hundreds of nuclei throughout it. Now, a muscle itself is just loaded with protein, mostly actin and myosin, which are the contractile units. We're going to get to exactly what those are and how that works in a bit. And the whole cell is just designed to contract. So why do these cells maintain all of these nuclei? Why after fusion, why don't the nuclei fuse somehow, or why don't they just go quiescent and get resorbed? So there is this structure that I'm giving you. There's a structure I'm giving you. It's a large cell with tons of nuclei in it. What's the functional significance of that? Can you guess? Intuit it. Think about it. What do you guys think? So how many athletes are in the room? Yeah, a good chunk of you. And what happens when you start doing some weight training? What's that? You can break down some muscle if you are pushing a muscle fiber to the point of it needing repair. Absolutely, you can. Before that, though, I'm not talking about putting yourself at the very limit. I'm just saying during general strength training, what happens to your body? When I was a kid, I used to read comic books, and in the back of the comic book, there would be these ads about wimpy kids at the beach, and they would get really bummed out because the like tough guys, the studly guys would kick sand in their face at the beach. And if you were like sick and tired of that, you could do the Charles Atlas program. And within like 6 months, turn yourself into this gleaming Adonis and not take sand in the eyes any longer. What happens when you start working out? You get bigger. You put on weight. Your muscles grow. And so I said that these cells are full of protein, right? Where does protein come from? Is it just born there? No, it's not. You guys know better. Where does protein come from? Amino acids, yeah. How do we put those together? Huh? Translation. What does that mean? Please translate it for me. Fantastic. Where does that mRNA come from? The nucleus. All right. So I just said these cells are big, right? Like there can be 30 centimeters. That's a big cell. Okay, that's a pretty big thing. And if there was one nucleus way over here on that cell that needed to grow the cell over on that end, it's kind of inefficient, isn't it? So in a cell that's this big, it helps to have nuclei dispersed throughout so that we can produce the necessary proteins throughout the cell more efficiently. All right. So there's this wide distribution of nuclei that allows the cell to grow and repair itself as needed. There's also a bunch of mitochondria all over the place because that requires a bunch of energy. So let's look here. We said it was voluntary, multinucleate, and striated. It can be as long as 30 centimeters. Does this picture give me anything? Well, it does give me this. We get to see how beautiful the striation is. And if we look, it's not just dark band, light band, dark band. There's like a fine pattern to it, right? There's a finer pattern to it. Well, all of that has a real significance that we're going to see. It's not just for looks. All right, so before we dive into it, though, I need to give you some anatomy. We have to do some cellular anatomy here. Okay. Oh, look at this diagram. A little overwhelming. A couple things. And let's start with the outside of the cell. The outside of the cell is called the sarcolemma. Sarco, I think I have it probably on another slide here, but I'll anticipate it by saying that sarco comes from the Greek sarx, S-A-R-X, which is the ancient Greek word for meat. and lemma is just layer. It's the layer it's the plasma membrane is what it is. It's the outer membrane of the cell, but specifically and only in a skeletal muscle fiber, the sarcolemma. And the reason we give it its own special name is because it is, can you guys guess? What do you think is going to be special about that membrane? Thinking back to what I talked about last week. What kind of membranes did I say there were? Say it. Excitable! That's it. Excitable membranes. Yeah, so the sarcolemma is entirely an excitable membrane. On the surface of them, you'll see throughout them these little pores. There are these little pores all over the membrane that communicate with this incredibly fine meshwork of tubules that go through the interior of the cell. We call them transverse tubules. Transverse because they cut transversely across the cell in this regular pattern. In fact, in a pattern that mirrors the striations that you see when you look at it in regularity. So these transverse tubules go in to the cell, and they surround these myofilaments or myofibrils. All right, not myofilaments. Myofilaments make up myofibrils. These myofibrils, which are bundles of proteins. Surrounding these bundles of proteins, we have a sleeve of another membrane delimited structure, specifically called the sarcoplasmic reticulum, just like the endoplasmic reticulum, except in a muscle cell. So we call it a sarcoplasmic reticulum. The sarcoplasmic reticulum is very similar to the ER, of course, but its primary function is to store calcium, because it turns out that calcium is going to be an important trigger in muscle contraction, and we'll see how that works in a bit. At the end of this sleeve of sarcoplasmic reticulum, there are these pouches, which we call terminal cisterna. Terminal, the end. Cisterna, a cistern is simply like a collecting. In general parlance, a cistern would be like a water well that would be filled with rainwater or something that you would drink out of. But these are just cisterns or wells that contain the calcium. And you'll notice that they're right next to these T-tubules, these collars of T-tubules. A T-tubule with two adjacent terminal cisterna, we call that a triad. We call it a triad. So a triad is essentially a terminal or a T-tubule sandwich on terminal cisterna bread. What else here? I think that's it. Yeah. The muscle fibers are red because they're packed with myoglobin. We'll talk about myoglobin later, but it's essentially a protein related to hemoglobin that stores oxygen at points of peak activity. We'll talk about that in a bit. Is there anything else? I don't think so. But I want you to understand, though, that if you were super teeny tiny, like you shrink yourself down, and you were walking on the sarcolemma, you could walk into one of these tubes and not leave the membrane. You're still on the membrane. And you would be able to walk through this complex network of tubules and still, in some ways, be on the surface of the cell. Okay? In the same way, like I think I've talked about being teeny tiny and walking down your mouth and being in your gut. You're still on the surface of your body, or an external access point, right? All right. So these myofibrils are the fundamental unit of striation, right? They get striated. They're striped, and this fundamental unit of striation is called a sarcomere. Sarx just means flesh, as I had said, and meros is part. So it's these the fleshy parts of the cell. It's these repeating units. The names of these, there's actually German microscopists that initially discovered all this stuff when they looked at muscle fibers quite closely. They gave them these different lines. the A band, the I band, the Z line, the M line, and then this zone of overlap. A band refers to anisotropic. Has anybody heard that word before? I'm missing my biochemist in the back row. How about isotropic? Probably not either. So isotropic and anisotropic, something that is anisotropic is the same in all directions. It does not have a handedness to it. There's not a directionality to it. And so what this is actually referring to is light. Light. If you were to shine polarized light, a lot of microscopes have polarized lights. If you were to shine polarized light on this sample, the A band, if you were to change the angle of the polarized light, the A band would not ever change the way it looked. However, the I band would, meaning it rotates light in a certain handedness. And we'll see what that means. I band and A band refer to how they interact with polarized light, but they do have, there's a molecular origin to that, which we'll see in a little bit. The Z line, you can think of it as standing for zigzag. You'll see what that means in a moment, but actually it stands for Zusammen. Does anyone speak German in here? No one? Nobody. Zusammen stands for adjoining, in between. Like something, Zusammen is between something. So it's the line between two adjacent sarcomeres. And then there's the M line. M line stands for Mittel, M-I-T-T-E-L, middle in German. So the Z line the two Z lines are the boundaries of a sarcomere, and the M line is in the middle. The zone of overlap, we'll see what that even means in a moment. Let's put a cartoon up instead now. There are these thick and thin filaments that make up a myofibril. These myofilaments, thick and thin filaments. A thin filament is going to turn out to be actin, and a thick filament is going to turn out to be myosin. But where there is only thin filaments, that's the I band. And A band is where we have thick filaments. The zone of overlap is where there are both thick and thin filaments. This H band down the middle is where there's only thick filaments. And the M line is the anchor point for all the thick filaments, and the Z line is the anchor point for all the thin filaments. There is this protein called titin, which helps all of these filaments stay in line. All right, and it also helps bring the muscle, keep all the myofilaments aligned with one another, and it helps with relaxation. So I showed a little picture of a titin monomer there. All right, so is this picture helpful at all? I always question it. I'm always about to delete this picture. But these are just cross sections through different points in the sarcomere. So you can see the kind of geometric array, the hexagonal array that these thick and thin filaments arrange themselves in a sarcomere. All right? So I'm just showing you the structure of a sarcomere first, and then we're going to talk about how that relates to its function. Yeah. All right. So first, thin filaments. Thin filaments are made up of actin. And a monomer of actin is called G-actin. It stands for globular actin. Globular actin is kind of like, in this diagram here, it's like the pearl on a chain. It's the pearl on a chain. And then a bunch of strands, a bunch of monomers of G-actin form this helical structure around a protein called nebulin to form what's called F-actin. So this whole strand is F, or filamentous actin, around the core of nebulin. Now, on the outside of this strand of F-actin is this these two proteins called tropomyosin and troponin. They're very important for the function of muscle contraction. So the first thing is tropomyosin. Tropomyosin is this also spiraled or helical protein that sits right on the surface of the F-actin and prevents its interaction with myosin. It covers an active site. There's an active site on the surface of the muscle that gets covered by tropomyosin. And troponin is the regulatory protein. It's the one that controls whether tropomyosin is held in place on the active site or whether it's going to be rolled away so that myosin can then interact with actin. The key to this is calcium. If calcium is present, then troponin will roll tropomyosin off the active site, and we're going to have contraction of the muscle fibers. If calcium is not there, then troponin will roll tropomyosin back onto the active site. Okay? So that's anticipating some stuff. We're going to go through it in much more gory detail in a moment. You guys got that? All right. On the other hand, there's thick filaments. So let's go back actually for one moment. If you look here, we see that there's a handedness to this spiral. This is why it's the I band. It's isotropic. All right. It like polarized light is going to hit that protein and get refracted in a certain with a certain directionality to it. All right. So it's going to bend polarized light uniformly in a certain direction. On the other hand, the A band, the anisotropic band, is from this myosin. So myosin is like a big bundle of little golf clubs with little putters with floppy heads. I don't know what else, how else to describe it. And imagine the putter is like two putters that have been where the handles have been twisted together. So here is a myosin molecule, and here's the head group. There's this hinge region, which actually isn't floppy the way I just said it was, but it is able to move, and it's the spring. It's more of like the spring that's going to cause this head to whip forward. All right. This is where the power of a contraction is going to come from, and we'll see how that works in a bit. But there's a whole bunch of these myosin molecules bundled together in this array that is a thick filament. And then the ends of it connect the thick filament to the Z line, the titin, the strands of titin, help with recoil. All right, so these are all the important players. There are some other proteins. Just having these filaments contract, and we'll see how that works in a bit, isn't enough to actually create muscle tension on the tendons that the muscle fibers find themselves in. There's all these other accessory proteins that help connect the sarcomere to the connective tissue outside, the endomysium, paramysium, and epimysium of the muscle. All right, and so they have all these different names. We're not going to go into it. It does not matter for our discussion. Except I was going to point out one of them called dystrophin. Dystrophin was named as such because it was discovered to be the protein that a mutation in which a missense mutation leads to muscular dystrophy. This disorder here where the muscle is not able to grow properly, and when the muscle does contract is not able to actually generate force on the tendons. Dystrophin, one of the linker proteins. Okay, so you'll have to forgive me for being a curious soul about how the universe, the greater forces that are at work in our universe, but I put this slide in just because it's an interesting thing to me, and perhaps it'll get you to think differently. I've talked a little bit. Have I talked about fractals at all in here? A little bit. People, do you know what that term means, or the notion of a scale invariant roughness, or looking at a pattern that repeats itself down a size scale? So, you can take a satellite image of the coastline and then zoom in, and there's repeating patterns, there's repeating motifs. Well, muscle tissue has a fractal element to it. There are these repeating motifs on different organizational levels in the muscle. So there's a skeletal muscle, which is a bundle of fascicles surrounded by epimysium. And then a fascicle is simply a bundle of muscle fibers surrounded by paramysium. A muscle fiber is a bundle of myofibrils surrounded by endomysium. And then a myofibril is just a bundle of myofilaments surrounded by a sleeve of sarcoplasmic reticulum. And then a sarcomere itself is simply a whole bunch of thin and thick filaments. Thin and thick filaments are themselves made of individual proteins that polymerize. All right, so there's this sort of like structural motif that moves down through the hierarchy, which I find to be kind of fascinating. All right, muscle contraction. An actual muscle contraction is simply an interaction between a thick and thin filament. I have 15 minutes, so I'm going to go through it. And on the Moodle, there's a bunch of interactive media. It turns out that when I put those media up there, they worked with all browsers, but in the meantime, the browsers moved forward, and they only work on Safari now. So if you want to watch any of those, you got to watch them on Safari. But there is one that shows you could spend 2 minutes watching the video and probably get 20 minutes of lecture out of it. It shows a little animation for how all of this works, how all the muscle fiber, what a muscle fiber contraction is on a molecular level. I suggest that you check it out. I'm sure there's other stuff on YouTube you could look at too, similar animations. So here's a little portion of a sarcomere that shows the H band, which is where there's thick filament but no thin, and then a little bit of the thin filament in the zone of overlap. And it's simply the motion of thick filament on thin filament that is contraction and is able to put force on either end of the muscle fiber. When you look at the muscle fiber, so all of this is going to require ATP, energy. And you look at the muscle fiber, you're going to see a bunch of mitochondria, but you're also going to see a lot of glycogen. Glycogen is animal starch. It's just a storage molecule for glucose, which is the unit, the sort of like $100 bill of energy in the body. And we're going to break glucose down to make ATP, which is kind of like the 20, I suppose. Those ratios are not exact. But you see the glycogen in green right here. I guess the mitochondria are in green, and the glycogen vacuoles are the ones in brown. Pardon me. All right, so let's look at the sliding filament theory. So we have a sarcomere at rest. We have the A band and the I band, and the zone of overlap is the size of it is. And when we get contraction, the A band stays the same size. You'll notice that the A band doesn't change in size. It's just that the I band gets shorter, gets smaller, because the filaments slide over one another. All right? Essentially, Z lines move closer together. Sarcomeres get shorter. Do you guys want a little visual demonstration of that? Should I get a couple of you out of your seat and do a little thing? Some volunteers? Are there any volunteers here? 1, give me 2, 3, Okay, 2. Give me one more. 3. Anyone else? No one else wants to get up? Come on up front. All right, so stand in a line right over here. And spread yourselves out. Stick your arms out. All right. And how is this going to work here? So now, Elizabeth and Zoli, you guys are thick filaments, and Molly and I are going to be thin filaments. All right. So we're going to just be passive, and they are going to be active, and their job is to do like this. When we contract, they're going to go like that. All right. They're going to pull along. They're going to pull along the arms of the thin filaments. All right. And we're going to see what happens. Give it a shot. So Zoli, grab Molly's wrist there. Spread out. Nice, well stretched out muscle. You guys ready? You ready, Elizabeth? Go for it. Go, Zoli. You hold my hand. All right. Ooh, yeah, we're contracting. See that? Look at that. That's amazing. That's all that it is. That's all the muscle contraction is. Did you guys catch that? Reasonably? Okay. Great. It's fun to hold hands for a minute. Okay. So here's an overview of it. There's going to be some kind of neural control that comes down through the neuromuscular junction. And then it's going to lead to excitation of the sarcolemma. That excitation is going to travel over the entire excitable membrane down through the T tubules into the interior of the cell, which is going to elicit calcium release from the calcium stores in the sarcoplasmic reticulum. That calcium is going to bind to troponin, which will roll tropomyosin off the active site on F-actin, and then we're going to go through a contraction cycle. So that what happens here at the sarcoplasmic reticulum is called excitation-contraction coupling. The release of that calcium is excitation-contraction coupling. Then there'll be the contraction, muscle fiber contraction, which will generate tension, and everybody's excited about excited membranes. All right, so let's go through it step by step. I just said there's the 4 major phases of contraction and relaxation. There's the excitation that happens at the neuromuscular junction. Then there's this excitation-contraction coupling that links what happens at the neuromuscular junction to what happens at the myofibril. All right. And then finally, the contraction phase, where there's actually the mechanism of myosin head group motion. And then finally, relaxation, bringing the muscle down to its resting length. So let's start with excitation. All of this happens at the motor end plate. So we see the synaptic bouton, synaptic knob of the muscle fiber, the motor neuron, full of all of these synaptic vesicles, and each of those synaptic vesicles has the neurotransmitter in it, acetylcholine. This acetylcholine is going to open up chemically gated sodium channels, all right, that are on the postsynaptic membrane. Here's the presynaptic membrane. Here's the postsynaptic membrane. We're not going to go through at the moment the detailed molecular mechanism of the release of all those vesicles. That's more neuro. But suffice it to say that that's the key. So step 1, action potential arrives at the synaptic knob. Here are all our vesicles of acetylcholine, and this action potential causes a degranulation of the acetylcholine. The acetylcholine is going to come over and open up these chemically gated sodium channels. Chemically gated sodium channels. Once that happens, we have the flux of ions Yeah, this is a redundant slide. Let's move to this one. Once that happens, we're starting local currents right here that are going to trigger these voltage-regulated ion gates. And these are the same characters that we talked about last time. They're the same characters we talked about last time. The sodium one is the depolarizing, and then afterwards, when you get all the way down, the potassium one is going to be the repolarizing gate. Okay? So the chemically gated one is the trigger, and then it starts the voltage gated one, which is actually what makes the membrane excitable. We're not showing the leak channels here. We're also not showing the sodium-potassium ATPase, the sodium-potassium pump, which also has to be there. Those are other players at the postsynaptic membrane. But all right, so this starts an action potential on the sarcolemma because this is excitable membrane, and then the acetylcholine just diffuses off the receptor. It diffuses off that acetylcholine receptor and gets bound by a protein called acetylcholine esterase, which is going to cleave an ester bond between an acetyl group and the choline. And both of those two things are going to get recycled. And the neuromuscular junction then returns to the initial state. All right. So we've started an action potential. We've knocked over the dominoes on the sarcolemma, on the membrane of the muscle cell, so to speak. So this action potential is going to travel across the entire membrane of the muscle cell, down into the T-tubules, and we're going to get ready for excitation, contraction, coupling. We got to take this depolarization on the membrane and translate it somehow into an actual contraction event. So this happens at the triads. The T-tubule, the depolarization goes along the membrane down into the T-tubule and causes the sarcoplasmic reticulum at the point of the terminal cisterna to release all of the sequestered calcium that's in this sarcoplasmic reticulum. There's suddenly this flux, this efflux of calcium into the sarcoplasm of the muscle fiber. Calcium floods the cytosol. All right. So now, the last step in excitation, contraction, coupling is that calcium is going to bind, I've already said this, but calcium is going to bind to troponin. When calcium binds to troponin, it rolls tropomyosin off the myofibril. So the way in your head you can keep troponin and tropomyosin straight is tropomyosin. It has myosin in its name, meaning it's going to interact with the myofibril. All right. Calcium binds, rolls the troponin tropomyosin complex out of the way from the active site, and now everything's ready for contraction. We can form a cross bridge between myosin and actin, and we can go through contraction. Who's had all this before? Any of you? Oh, good. A couple of you. What was the point I was making? T, tubules, and circle. Yeah, okay. Right. So now we're on to the contraction cycle. We've done everything that we need to get ready to contract, but we haven't had a contraction yet. The first thing that happens is we start with the resting sarcomere. There's the troponin on the active site. The heads of the myosin are cocked back and bound to ADP and inorganic phosphate. Calcium binds, revealing the active site, and that allows cross-bridge formation. Now, let's not go there. I don't have time for it. There's this cross bridge formation. Each step in contraction causes a conformational shift that directly relates to the next step. All right, so indeed, contraction is again like a set of dominoes. It's like a set of dominoes. So each step that happens leads to a conformational shift that the direct consequence of is the next step in contraction. You guys with me? Okay. So as the cross bridge is formed, as that myosin head contacts the actin, it causes a release of ADP and inorganic phosphate. ADP and inorganic phosphate cannot remain bound to the head group after cross bridge formation. When they are kicked off, ADP is specifically the thing that's keeping the head group open. As ADP falls off the head group, the hinge, the spring in the hinge springs. This is actually contraction. This is the moment of contraction right here. All right? All right, so I think I'm at time. So I don't like letting you guys go over. Yeah, let's call that time, and we'll pick up right here. I got you right to the moment of contraction, and we'll take it to the finish line next time on Friday. On Wednesday, quiz on Friday. Thank you. Any questions, comments, expressions of enthusiasm or dismay? No? Okay.